Hello, in this video we will discuss about the cholesterol synthesis and their cholesterol formation, and we will understand about the what is cholesterol. So the cholesterol synthesis occur only in the liver cells and basically hepatocyte, and cholesterol is derived from fat. That is why cholesterol fat soluble, and cholesterol produces steroid hormone, for example estrogen, testosterone and progesterone and dihydrotestosterone. This is a male and female sexual hormone which that appear and to produce a sexual behavior, the secondary and primary sexual character. While the cholesterol can be come from the food, for example egg, milk, vegetable, fish and other sources, and the little bit amount of cholesterol will come from the food, and in this way the high cholesterol is come from the fat from the by the synthesis into the liver. So here is the formula of the cholesterol. You can see that is a four ring like structure and the carbohydrate ring. So the carbon chain ring. So here I am drawing the liver and blood vessel, blood circulatory system, and here is the digestive system, which is made up of stomach and duodenum I am drawing here, and the pancreas. So remember about that. When we will take the food that is go into the stomach, and after the digestion from the protein and further go into the duodenum, where the duodenum is basically contain a pancreas, and pancreas contain a exocrine hormone, exocrine enzyme, sorry. So this exocrine enzyme is the pancreatic lipase, as well as the gallbladder will produce from here is the bile pigment and bile salt for the emulsification of fat, and the lipase is used for the digestion of fat, will convert the chylomicron, which that is the very low density lipoprotein, which contain a less amount of the protein while the high amount of the lipid, and also cholesterol can be from the dietary cholesterol. So the lipase enzyme used to deposit the cholesterol as well as lipids into the adipose tissue as well as other tissue. So the remnant cholesterol, the remnant chylomicron will be absorbed via endocytosis into the liver through the scavenger receptor and go into the cholesterol, and extra cholesterol will be stored into the gallbladder. So what that means? That means the extra cholesterol is not harmful. Why? Because it is excrete from the into the gallbladder. So here the function of the cholesterol is the plasma membrane of the cell, which that is used for the integrity of the plasma membrane and the fluidity and the stretching of the membrane for absorption of water, etc., will turgid and will not, and stability will be occur. So the water permeability will be decreased due to the cholesterol, due to the hydrophobic and hydrophilic interaction. So here is the liver is the major organ for transportation of lipid to the cell membrane. So the glycogenesis first of all and glycogenolysis will be occur when food will not intake. So the glycogenesis means the glycogen formation, while the glycogenolysis is the breakdown of the glycogen into the sugar. While the high density lipoprotein will be excreted from the liver to go into the several tissue, while the low density lipoprotein will be enter into the liver. So that is a general concept. So let's begin to understand the liver hepatocyte. So the hepatocyte general map we will discuss about the synthesis of cholesterol from the liver. It is the cholesterol is the non-essential dietary compound which that is come from the fat. So the fat is the essential part of our body, for example. But the fat is also produced from the sugar we will discuss in the next topic. So here is the glycolysis will be occur when the sugar will be entered through a GLUT transporter channel 4, and this convert into the pyruvate, and this pyruvate will go into the mitochondria, and after the go into the mitochondria the Krebs cycle preparation and electron transport chain to produce acetyl-CoA. This acetyl-CoA will efflux of the acetyl-CoA to convert the acetyl-CoA from the 2-acetyl-CoA, and the HMG-CoA will synthesize cholesterol. So the malonyl CoA is produced from the fatty acid, that is the basically the malonyl CoA. This malonyl CoA come from the beta oxidation, and that is basically you can see and produce the malonyl CoA produce the monoacyl glycerol by the attachment of one fatty acid and the second the diacyl glycerol and triacyl glycerol. This triacyl glycerol will be packaged into the lipoprotein uh, lipoprotein lipoprotein globule. So let's begin to understand the biosynthesis of cholesterol. Step of synthesis we will discuss here. So the acetyl CoA will be converted into the 3-hydroxy 3-methyl CoA, and it is known as the HMG CoA. And this HMG CoA is used for the conversion in the presence of HMG CoA synthase enzyme. While here is the acetoacetyl CoA is also produced from the acetyl CoA, and this acetoacetyl CoA will convert into the triacyl glycerol or go into the 3-hydroxy to produce HMG CoA. While the HMG CoA synthase enzyme used to produce the mevalonic acid, while the inhibitor is the statin, which that is the HMG CoA reductase. It is the statin which that is inhibit the production of cholesterol from this mechanism. While the mevalonic acid in the present produce and the ATP used to mevalonate kinase enzyme to produce the mevalonate 5-phosphate, and this mevalonate 5-phosphate convert in the presence of mevalonate 5-phosphate decarboxylase enzyme. So the carbon dioxide will be released and the produce isopentenyl 5-pyrophosphate, and this is the pyrophosphate isopentenyl, and the isopentenyl pyrophosphate isomerase enzyme to produce the dimethyl allyl pyrophosphate, and the farnesyl pyrophosphate synthase or biphosphonates will used to produce the geranyl pyrophosphate, and this geranyl pyrophosphate is converted in the presence of farnesyl pyrophosphate synthase or again this is the used to produce the farnesyl, and this is the farnesyl basically is the synthase. So in this way you can see and understand to produce farnesyl pyrophosphate, and the squalene synthase enzyme used to produce squalene, and the squalene monooxygenase enzyme used to produce 2,3-oxidosqualene, and this squalene epoxidase enzyme used to produce the epoxidase to produce the by using of the NADH to produce the lanosterol. So lanosterol we convert in the 19 chemical reaction to produce cholesterol. While here is the farnesyl pyrophosphate we convert into the geranyl pyrophosphate. While the geranyl pyrophosphate can be converted into the prenylated proteins, and on the other hand, farnesyl pyrophosphate is also converted into the heme A polycol ubiquinone, and on the other hand the cholesterol will be deposited into the high density lipoprotein inside, and in this way the esteration will be occur. So the cholesterol is in the form of ester bond with the so here is the phospholipids surrounding. So this HDL will be excreted from the liver to use into the several tissue for deposition of fat, deposition of protein, and deposition of the cholesterol. So in this way, this is the mechanism of the biosynthesis of cholesterol step of the synthesis. So that is the cholesterol, basically the overview of cholesterol.